Right, so we're gonna keep moving on with our genetics and how traits are kind of passed along. So we're starting on page 36. And just kind of a quick note on connecting pedigrees to alleles. Just remember you've got alleles, so that's kind of your two traits. So as an example, we have, say, GT, or if we're looking at blue eyes, have your two alleles, two small b's. So we can add alleles to family pedigrees. So on like any potential quizzes, the alleles would kind of deal only with complete dominance and not necessarily recessive traits or other types of dominance, which I'll talk about later in this video. So with your family pedigrees, just want to double check that you did list alleles on there. So based on the traits in your family, should be able to try and figure out who's who. You might not always be able to figure it out. So some might just be a question mark depending on the traits and which trait you're tracing. So, but just again, like the trait that you're looking at can be dominant or recessive. So we're only looking at like simple dominance with these pedigrees. You kind of have to figure out whether or not the trait you're looking at is dominant or recessive. Got kind of a couple of examples that can kind of show how this works. So that mini pedigree on the left have two unaffected parents, and their child is affected by the trait. You can see that both parents would be carrying a recessive gene, and their offspring has the two recessive alleles. So this would be a recessive trait. If you do see the trait being passed along, regardless of, if you do see the trait being passed along, so this example we have got a pair with its homozygous dominance and a homozygous recessive parents. That offspring, again, could be carrying, or at least has a one dominant allele, and then that dominant allele gets passed along. So with this mini pedigree, we see a dominant trait being passed along. All right, so now we're going to look at a few other types of dominance. So kind of looked at kind of like fur colors or eye colors or like widow's peaks or kind of attached or unattached earlobes as some different examples of complete dominance. So it's if you have the gene, or if you have the dominant gene, you will express the traits. If you have the two recessive genes, you won't express it. Those are just examples of complete or simple dominance. Now we're going to make it a little more complicated and look at a few other types of dominance. The first one is called codominance. So both alleles are expressed. So like they are codominant, or say, imagine a co-worker working together. So we have an example here of a chicken. So with these codominant alleles, you could still drop Punnett squares for them. But we're going to kind of change a few things and add in some superscripts. These codominant genes, so we're at the chickens, we're looking at feather colors. So we use F for feathers. So it's kind of a big capital F to start out your alleles. And then you're going to write down a different superscript depending on what the allele is. So if you have a all black chicken, again, there you have your F, then the superscript B, F for feathers, so then superscript B, again, B for black. On the other side, if we have an all white chicken, again, we're still using F for feathers. Then we use two superscript W's to show that it's a white chicken. For a checkered chicken, it has both alleles. Both alleles are being expressed, so it has kind of a checkered pattern, black and white. And we're still going to use F for feathers. One allele will be B for black. One allele will be W for white. So we've got an example of kind of breeding some chickens. To kind of explain, like heterozygous have black and white feathers. Homozygous, it's either going to be all black or all white. So we're going to show a breeding of a black chicken and a white chicken. So we can still draw our Punnett square. So again, just F for feathers and black, and FW for the white chicken. All right, so we just draw our Punnett square as normal. Copy the alleles down for each parent, down or across for each parent, and we can kind of fill it in. So we could still figure out our genotypic and phenotypic ratios. So with this genotype, all of the offspring will be FB, FW, so 100% checkered for the phenotype. For next example, we have a checkered chicken breeding with a homozygous white chicken. So again, we follow the same rules of the Punnett square. So FB, FW on top, FW, FW on the side. End up with a genotypic ratio of 50% checkered, or 50% FB, FW, 50% FW, FW, and a phenotypic ratio of half checkered, half white. So this isn't the only example of codominance. Got a few others. So horses, if you cross a chestnut horse and a white horse, you end up with a kind of roan-colored horse. Some flowers, cross like a pink flower and a white flower, you end up with a flower that kind of has mixed petals. So some petals will be pink, some petals will be white. In your notebooks, you have another example of this thing called sickle cell anemia. So it could be codominant with kind of regular blood cells. Right, so on the next page, there are a few examples of Punnett squares that you can work through. Pause here, take a couple of minutes, try and fill those out. All right, so hopefully you're able to figure those out without too much trouble. I'm just gonna kind of show the answers to you. You can kind of compare your work. Make sure you got your Punnett squares filled out properly. If you have questions, let me know. If something's kind of didn't work out, again, let me know. Again, just kind of take a second, compare your answers, and kind of keep on moving, and we'll look at another type of dominance, complete dominance. So this is when one allele is not completely expressed over another. So kind of like blend together. Neither one's dominant over the other. So these types of dominance kind of follow similar rules to codominance. So you use one big capital letter for the gene. So then again, like C for color, or if you're doing chickens, F for feather. And we're still going to use a superscript for the individual allele. got a few examples that we're going to look at with dealing with carnations, type of flower. So we're going to use like C for color, R for red, and W for white. And there are kind of like some references out there that just use an R and a W when kind of showing incomplete dominance. This is why it was really important for you during your Skittles lab not to kind of mix colors up with your Skittles. When you're looking at simple dominance, you always want to keep your traits as a uppercase letter and a lowercase letter, so you don't mix it up with incomplete dominance or codominance. Right, so with carnations, again, a type of flower. Homozygous dominance would be CR, CR. Homozygous recessive would be CW, CW. In this incomplete dominance, you'd have one of each allele, and you'd end up with a pink flower. Again, we've got another example of a Punnett square where we're crossing two pink flowers. So we have one pink flower, CRCW, another pink flower, CRCW. Cross them following the same kind of crossing rules we've been following with Punnett squares. And we can still calculate some genotypic and phenotypic ratios from that. So with our genotype, we end up with 25% CRCR. Half of them are CRCW, so half will be pink. And 25% CWCW, so 25% or a quarter will be white, and the remaining quarter will be red. Again, we've got another example. So this time we're crossing a red flower and a pink flower. Going to end up with some different ratios than with the two pink flowers. So we're crossing a CRCR and a CRCW. Half of them will be red. Half of them will be pink. In your notes, you've got a few other examples of incomplete dominance. So just kind of cross out where it says codominance, place it with incomplete dominance. One possible example of incomplete dominance is some breeds of mice. So you have a black mouse, a white mouse. They breed, you could end up with a gray mouse. They kind of blend together. A human example is kind of with hairstyles. So if you have curly hair and you have straight hair, you could possibly end up with wavy hair. So that those two types kind of blend together. On your next page, you've got some more Punnett squares. Kind of work through those. Practice your incomplete dominance. So just kind of pause, take a second to complete those. So we've got two of these questions kind of answered. Double check your answers. Take a look at 3 and 4. Kind of work through them yourselves. If you have questions about these two, let me know. All right, so got a QR code and we've got a link here. I'll post this link down in the description so you can go to this website. This is kind of a quick color blindness test. So I'll let you try this test right now, and then I'll come back to it when we get to our sex-linked traits. So just take a few minutes, try out this color blind test. You might be surprised, you might not be. And we'll kind of keep moving on once that's finished. Right, so the reason we tried out that colorblind test is because it is a thing called a sex-linked trait. So these are traits that are only located on the X chromosome. So color blindness, where you have some missing cones in your eyes, so you can't distinguish certain colors as easily, is a mutation that's occurred on the X chromosome, not on the Y chromosome, only on the X. So this matters when we're kind of looking at the X and the Y chromosome. So the Y chromosome is quite a bit different than the X chromosome. Any alleles on the X chromosome will always show up in males, whether or not it's dominant or recessive. It's because the Y chromosome just does not have space to have kind of matching alleles on it. It's a much smaller chromosome. And if there's a recessive trait on the X chromosome, there's nothing on the Y chromosome that could potentially block that trait. So females are much less likely to be affected by a recessive X-linked trait, because since it's recessive, they need 2 copies of it. Males only need 1 copy of it. However, they can carry this trait and potentially spread it or have an affected male offspring. So there's a couple of really common sex-linked disorders. One is color blindness. So color blindness is just the inability to distinguish the difference between certain colors. So the most common one is red-green color blindness. So red and green are seen as almost the same color. So we've got a picture here of some dots. It's a common color blindness test. If you are colorblind, all of those dots would look to be the same. If you are not colorblind, you would see a number inside of those dots. So you can still draw Punnett squares for these sex-linked traits. So again, we kind of follow similar rules. So we've got X, we've got Y, we've got X and X. So for example, if we're using color blindness, say an X with a capital N for normal, X with a lowercase n for that recessive allele. And if we're looking at, could still match it up with the male XY, but notice that there's no allele attached to the Y. There's just kind of nothing that will match up with that allele on the Y chromosome. So just don't write anything there. We've got a Punnett square, kind of showing a cross between a female carrier and a normal male. So the female has normal vision, but she carries the color blindness allele. And we just kind of want to figure out the expected phenotypes of the children. So we can still give our normal genotypic ratios and phenotypic ratios. These are going to be a little bit longer than normal. Right, so for our genotypic ratio, we kind of end up with a 1:1:1:1 ratio. For our phenotypic ratio, we'd kind of end up with 75% normal or normal vision, 25% being colorblind. Other common sex-linked traits is this thing called hemophilia. So hemophilia, again, carried on the X chromosome. It's basically a genetic disease that occurs when blood does not clot properly. So if you get a small cut, it will just keep bleeding. Like the bleeding doesn't really stop. This particular trait has had a pretty huge historical impact. So there's one really famous carrier of hemophilia, Queen Victoria. She did not have hemophilia, but she did carry the trait for it. Queen Victoria was around in kind of mid-1800s. And she's kind of an ancestor of a whole lot of European monarchy. Because of that, she kind of spread hemophilia throughout a bunch of the monarchs across Europe. It's kind of turned out to be pretty significant in Russia. So one of the Tsar Nicholas's children had hemophilia. He was one of the, I believe, either a grandson or a great-grandson of Queen Victoria. And that kind of led to his family trying to find a cure to his hemophilia, which led to a character named Rasputin, which kind of was a pretty significant cause of the Russian Revolution. But there were many monarchs and kind of members of royal families in Europe that ended up with hemophilia. Kind of became known as the royal disease. And again, we can still kind of work through a Punnett square. In this case, we have a normal female. She's not a carrier. She does not have the gene. Marries a male with hemophilia. All of those offspring will be normal, but all of the female offspring will be carriers of hemophilia. So in this particular case, the children will not have hemophilia, but there is a chance that grandchildren will have it. So we've got another example of sex-linked traits. Kind of occurs with calico cats, or cats with 3 fur colors. Only female cats can be calicos. That's black, orange, or black is on one chromosome, orange is on another. You need both of those to kind of show all 3 colors. And white is controlled by other genes, not on the X chromosome. So you just need those 2 X chromosomes to kind of see both or all 3 of those fur colors. All right, so again, we've got a few more Punnett squares to work through on sex-linked traits. If you have questions on these ones, let me know and you can work through it in the meeting. All right, so we're going to kind of wrap this up with these multiple alleles. So most common multiple allele deals with blood types. You might know that there are kind of 4 common blood types: A, B, AB, and O. So this matters if you need blood transfusions. If you donate blood, they're going to test it and see what blood type you have. Can't always receive any type of blood. It does matter which type you receive, but it doesn't always have to be the exact same as yours. So we'll talk about why that is. Remember back in grade 8, have these things called antigens. Antigen is basically a protein on the outside of kind of cells or microbes. It's a protein that your body does not recognize and it wants to destroy. So an antigen can trigger a response by antibodies. With blood, there's 2 different genes that kind of govern blood types. So humans, possible blood types can be, or possible blood type alleles can be listed as IA, IB, or a lowercase I. It's kind of similar to our codominance and incomplete dominance, where we're using the capital letter. We're dealing with blood types, we use the letter I. So we use I instead of B because I is short for isoagglutinogen. So it's kind of another word for antigen. With our blood types, IA and IB are both dominant, and our lowercase I is recessive. So you can end up with some either simple complete dominance of one, or homozygous dominance of one type, or we can end up with something kind of like codominance, or we can end up with kind of homozygous recessive trait showing up. Basically, each allele is kind of responsible for producing antigens on the red blood cells. Also kind of responsible for producing antibodies that'll attack foreign antigens or antigens the body does not recognize. So the alleles don't produce antibodies that are going to attack the body's own antigens. So that's why if you have type A blood, you can be given more type A blood. Antigens are the same, so your body's not going to attack them. With type I, so that's the recessive blood type, there's no antigens or antibodies on those blood cells. Since there's no antigens on it, there's nothing that the body will recognize as an invader. So the blood type O can be given to everyone. It's kind of known as the universal donor. We just kind of have a breakdown of those 4 blood types. We have AB, and O. A and B both have their own unique types of antigens. Blood type AB has both types of antigens. And blood type O has no antigens. However, if you have blood type O, it's going to kind of spot those A and B antigens, and it will kind of attack them, recognize them as invaders, and cause an immune response. So if blood type A, B, or AB is given to a blood type O person, there will be an immune response in that person, and it will not be good. So we just kind of have this table of different alleles, antigens, and antibodies. You can just remember that O is recessive. So if there's an A or a B allele in that person, just have that dominant trait expressed. So we've got one more Punnett square in the notes. So here we are crossing, got someone with AB blood and someone with type O blood. Again, we're using I for our isoagglutinogen or antigen. So IA and IB, then II since O is recessive. Can just kind of do a simple cross there. And we'd find out that the children could only be blood type A or blood type B. All right, and got another factor to worry about with blood. It's called the Rh factor. So it's kind of added onto blood types. So there's another antigen on the surface of blood cells. So I believe this one follows kind of simple dominance rules. So if people have this Rh antigen on them, they're Rh positive. If they don't, they're Rh negative. So this way we kind of double our amount of blood types, so we can end up with like AB positive, AB negative, O positive, O negative. And it's kind of another factor to worry about when donating or receiving blood. If you're O negative, you don't have that Rh factor, and you can donate to a positive person. However, a person that is Rh negative cannot receive blood from a positive donor. So this Rh factor does matter with pregnancy. It's possible for a mother to be Rh negative, and she could be pregnant with a fetus that's Rh positive. So Rh negative mother cannot receive any of those possible antigens from the fetus. Some of them can kind of cross the placenta. Again, that can trigger an immune response. So the mother's body would start to attack the fetus. It used to be a very common cause of miscarriages. For now, it's very common to kind of receive a vaccination to suppress this immune response during pregnancy. So this has kind of helped out many families. Right, so when we are kind of looking at this Rh factor, we end up with like 8 possible blood types. And here just kind of have a big general distribution of blood types. Kind of do vary with groups, across countries, different ethnicities. So the most common blood type is O positive. Least common blood type is AB negative. Again, it's not even across the world. Some areas are going to have higher concentrations of a certain blood type. Other areas will be much lower. Here, you just kind of have a kind of chart showing what you can give and what you can receive. So if you are O negative down at the bottom, you can only receive O negative blood. But if you're AB positive, you can receive blood from anyone. So we have the universal donor, and we have the universal acceptor. Right, so we're got a few more Punnett squares for you dealing with multiple alleles. I will let you practice on those as well. Have another assignment posted up for you on Teams. Meeting will be open, so if you have any questions, you can pop in and let me know. And send me a message if you're kind of stuck on anything.